Allora, grazie per questo ritorno relativamente sollecito. Siamo alla seconda parte del convegno che riguarda il genome editing, almeno come argomento generale. Il primo a parlare è il professor Ballabio, che tratterà del nuovo ruolo che è stato compreso dei lisosomi. Professor Ballabio will speak about lysosomes as a center of regulation of a metabolism, cell metabolism. Please.
Buon pomeriggio. Io ringrazio gli organizzatori del meeting, il dottor De Luca e il dottor Giuliani, per l'invito a partecipare a questo meeting così interessante e anche così eterogeneo. Io continuo sul tema dell'eterogeneità perché vi parlerò di un argomento in cui c'entra sì la genetica, ma c'entra tanto anche la biologia cellulare. Parlerò in inglese. Sono abituato a parlare in inglese, quindi vi chiedo perdono, ma anzi lo faccio anche in rispetto dei illustri speakers stranieri che mi hanno preceduto. Okay, so when I get this kind of invitation to meetings with such a heterogeneous audience, I always have the dilemma: shall I give a very general talk, or shall I focus on my research? And then I think and think and think, and then I always decide in the same direction. I speak about my research, because that's what I like to do. But I try to do it in such a way that people with different backgrounds will get interested. I don't know if I succeed, but I'll give it a try. So in my laboratory for pretty long time now, we've been focusing on a very special cellular structure, a little organelle called the lysosome. And so all cells, as you know, most of you are biologists, of course, and so I'm sure you know that all cells of our body contain hundreds of these organelles called the lysosomes, and you can easily visualize the lysosome by simple immunofluorescence using, for example, a lysosomal membrane marker. In this case, the marker is called LAMP1, which is a lysosomal membrane protein, and you can visualize these vacuoles that are present in the cytoplasm of all cells. Now, the lysosome have been discovered by a Nobel Prize winner who also discovered the peroxisomes. He also made many other important discoveries. And this is Christian de Duve, who won the Nobel Prize for Physiology and Medicine in 1974 for his discovery of the lysosome. Now, the lysosome is mostly known for its function in degradation and recycling processes. So its importance for the cleaning up of the cell. Now, the substrates that are degraded by the lysosome, they reach the lysosome in different ways. Substrates that come from outside the cells, they reach the lysosome through the endocytic pathway, so mostly through endocytosis. So they are internalized, and then they get into late endosomes, and then by a vesicular fusion, the fusion of two vesicles, they reach the lysosome. Now, also phagocytosis is another process, and so in this case, they are internalized, the substrate, they go to nascent phagosome, and again, by fusion, they reach the lysosome. And so these are extracellular substrate. Now, an emerging new process that's been studied by more and more scientists, there's even a journal now on this process, is the autophagy pathway. And so this pathway is needed by the cells to digest, to internalize into lysosome, to take to the lysosome intracellular substrate. It's called autophagy because it's a kind of a self-eating of the cell. Now, the discovery of autophagy also ended up in a Nobel Prize winner. This was very recently, 2016. Yoshinori Osumi won the Nobel Prize for Physiology and Medicine for its discovery of this pathway and many important genes that are important for the formation of this type of vesicle, which has a double membrane, which is the autophagosome, that fuses with the lysosome and delivering intracellular substrate to the lysosome. And intracellular substrate can be of different kinds, so aggregate proteins, also dysfunctional organelles, mitochondria, dysfunctional mitochondria, they can end up in the autophagosome and then get to the lysosome for degradation and recycling. So the main components of these organelles are of different types. Of course, this is a complex organelle, so it's made by many different proteins. So first of all, the lysosome has a membrane, which is very important because it separates the luminal environment from the cytosolic environment. And the luminal environment of the lysosome, as I'm sure you know, biologists, you know, it's acidic. And so this separation is very, very important. Also, there are membrane proteins, and they handle the traffic of material in and out of the lysosome. Sorry. Then there is a proton pump that is important for the acidification of the organelle. This is called the VATPase. I don't know what I'm doing. And then there are luminal proteins. Mostly these are enzymes, hydrolytic enzymes, that are needed for the degradation of specific class of substrates. So they have a different substrate specificity. Also, of course, inside the lysosome, in the lumen, you also find the material to be degraded. And a new class of proteins, which is very interesting, and it is something also that we are focusing on, are the proteins that get associated to the lysosomal membrane in a dynamic way. So they're not always there, but they get to the lysosome in some conditions, and their recruitment to the lysosome is important for their activation. And I'm talking about really important proteins and really important functions. So because of its role in degradation and recycling, the lysosome has always been referred to as the cell trash can. Okay? Actually, even the DUV itself defined the lysosome, I don't know if you heard about this terminology, suicide bag, because of this active role in degradation and recycling. Gli spazzini cellulari, basically. And also when people talk about the lysosome, they talk about the end of cellular catabolic pathways, and so the one who does the dirty job. And it's always depicted at the end of the slide or at the end of a pathway. The last thing is the lysosome. And then things get to the lysosome, and that's the end of it. Also, this organelle has been also referred to as a static organelle, so not really subject to regulation and not influenced by environmental cues. And all the genes encoding for lysosomal proteins were thought to be housekeeping, so not really subject to regulation by environmental conditions. And we thought this was not, well, I also added that some people even think that the lysosome stinks, which is, of course, a joke. But anyway, so we challenged this view. We thought, in fact, that the lysosome is much more exciting than that. And we postulated that the lysosome is a dynamic structure, so a dynamic organelle which is able to adapt to environmental cues. And we also postulated that lysosomal function is subject to a global transcriptional control through a master regulator. So we thought that the cell may need to increase or decrease lysosomal function according to environmental cue. And in order to do this, of course, to increase the function of an entire organelle, you need to increase the levels and control several components of the function of the organelles. You need to control the proton pump, you need to control the membrane proteins, you need to control the hydrolases, and the only way the cell has to do this kind of global regulation is through master regulators, which are genes controlling the function of many other genes. So we tested our hypothesis using in silico approaches, basically. So this was done a few years ago. So we postulated that genes and the expression of genes encoding for lysosomal protein will go up and down in a coordinated fashion. So this means co-expression. We thought genes encoding lysosomal proteins were co-expressed. So when the level of one would go up, we will also find the other genes whose expression will go up. And we tested this by analyzing transcriptome data, which were deposited in the databases and were obtained from different type of experiment performed by others, not by us. So we actually were looking into databases, transcriptome data from cancer cells, from cells treated with drugs, from cells from patients, and we would see perturbation of gene expression. And we would look at lysosomal genes, and their expression was coordinated. They would go up in some condition, down in some other condition. So there was significant co-expression, and co-expression very often is a sign of co-regulation. And so then we analyzed the promoter of the genes encoding for lysosomal proteins, and we found that many of them had, in their proximal promoter, had a typical signature, which is a known target site, a palindromic sequence, which is a known target site of a family of transcription factor called HLH leucine zipper transcription factor, and in particular a subfamily of the larger HLH family of transcription factors. So putting together the co-expression data and the promoter analysis data, we identified a new gene network which we named CLEAR, which is in fact coordinated lysosomal expression and regulation. But of course we named it CLEAR because we like this term. So then we identified the master regulator of the CLEAR network, and this gene is called TFEB, transcription factor EB. So this gene is a transcription factor, of course, encodes a transcription factor, and it binds to the promoter of the gene belonging to the CLEAR network, which, by the way, are not only genes encoding for lysosomal proteins, but then, as we will see, also many other pathways. And by doing so, this transcription factor promotes lysosomal acidification. It promotes the activity, the levels of lysosomal enzymes by increasing the expression and increasing the levels. But it also, amazingly enough, this gene also controls the number of lysosomes. So if you overexpress this gene, you get many more lysosomes. And of course, loss of function experiments will do, you get opposite effects. So this gene belongs to as I said, to a subfamily of HLH leucine zipper transcription factor family, together with 4 other members. They are very similar to each other. They also have a similar binding pattern to the promoters, but in fact they have a different physiological function because of different expression patterns. MITF is mostly expressed in pigmented cells, while TFEB and TFE3 are ubiquitous, and TFEC actually has very, very low level. So we don't know the role of this gene. So after the discovery that this gene controls lysosomal biogenesis, we also discovered that this gene is involved in autophagy, not only through the lysosome, but also by directly regulating some crucial genes that control autophagosome biogenesis. And this actually makes perfect sense, because in order to achieve degradation and recycling, not only you need to control the degradation process, but also you need to control the cargo delivery of substrates to the organelle. So So this was really, it's clearly a functional network. So you control the delivery of substrates by autophagy, and you control the degradation by the lysosome biogenesis. Now, this discovery became very popular because several people, several groups, including us, tried to use this master regulator as a way to promote cellular clearance in a number of different diseases. And I summarized some diseases for which this approach has been used basically by viral-mediated gene transfer in mouse models of a variety of disorders. The most obvious application was lysosomal storage diseases. These are terrible diseases caused by defective degradation of the lysosome. And so if you overexpress TFEB in these diseases, you rescue the phenotype by promoting cellular clearance. But several groups also tested it in Parkinson's disease, Alzheimer's disease, Huntington disease, but also other diseases such as alpha-1 antitrypsin deficiency and many others. Slower, okay, for the interpreter. Okay, then, but even in diet-induced obesity, we actually found that a profound effect, beneficial effect of the overexpression of this transcription factor. You see when you feed mice with the high-fat diet, you get a very significant increase in lipid droplets, and then, of course, you get obesity and diabetes. And this is where the liver, when you overexpress TFEB, you promote clearance, complete clearance of lipid droplets, and you rescue the obesity and the diabetes. Okay. So another fascinating aspect about this master transcriptional regulator is how its function is regulated. And so we discovered that this transcription factor, it's located predominantly in all cell types in the cytoplasm. Now, this is a transcription factor, so its activity should be exerted in the nucleus. But in fact, this protein is predominantly in the cytoplasm. And this is in normally fed cells and unstimulated cells. But then, according to a variety of conditions, particularly nutrient deprivation, both in cell culture but also in vivo, for example, we tested it in mice but also in humans, with nutrient deprivation, so with starvation, this transcription factor moves from the cytoplasm to the nucleus. So it gets activated by nutrient deprivation. So, and it moves from the cytoplasm to the nucleus. And now we and many other groups discovered that there's many different stimuli promoting the nuclear translocation. As I said, starvation, but also infection by bacteria and parasites, different type of stress conditions like ER stress, but also in the muscle physical exercise. With physical exercise, it moves from the cytoplasm to the nucleus and it gets activated, and other conditions. So, so how is this shuttling between the cytoplasm and the nucleus regulated? And we discovered that this is regulated by phosphorylation. So this is a post-translational modification. So when TFEB, this transcription factor, is phosphorylated, particularly in these three serines, then it's kept in the cytoplasm. When it gets dephosphorylated, it moves from the cytoplasm to the nucleus. So phosphorylation, in this case, it has an inhibitory effect. And of course, the next question was, what is the kinase that phosphorylates TFEB? And then here we and others made the discovery that a very, very important kinase complex, the mTOR kinase, in particular mTOR1 complex, phosphorylates TFEB. Now, mTOR is one of the most important hubs that controls cell growth and proliferation. So it's the kinase that controls cell growth and proliferation, mTOR, controls the master regulator of cell catabolism, TFEB. So this was kind of interesting. And mTOR, as you can see here, there's so many pathways regulated by mTOR. mTOR is regulated itself by a number of environmental cues, particularly nutrients and amino acids in particular. And then mTOR regulates autophagy, mRNA translation, aerobic glycolysis, de novo lipid synthesis, and so on. So look what happens when we treat cells with an mTOR inhibitor, one of the most commonly used mTOR inhibitors. It's called taurin-1. So this is a cell that is transfected with TFEB GFP. So the green that you see here is TFEB, basically. And you can see that in normally fed cells, TFEB is cytosolic. Okay? But then when you use the mTOR inhibitor, then you can see that it moves quite rapidly and quite steadily from the cytoplasm to the nucleus. And now there are several groups, including ours and several companies, trying to find drugs that, like this mTOR inhibitor, promote this nuclear translocation without having side effects, of course. Because this is a way to activate this transcription factor by using chemicals. So, actually, I'll very briefly, the other interesting aspect of the relationship between mTOR and TFEB is the fact that mTOR, to be active, needs to be recruited at the lysosomal surface. It's one of those proteins that we can call lysosome-associated proteins. To be active, mTOR1 needs to get on the lysosomal surface. So now we have a kinase that, to be active, needs to be at the lysosome that regulates a transcription factor who is involved in the transcriptional regulation of lysosomal genes. So this is clearly a sign of feedback mechanism. So during the anabolism situation, where the cell is full of nutrients and needs to promote protein synthesis, then mTOR1 is active, TFEB is phosphorylated, and so it's kept in the cytoplasm, so it's inhibited. But then with starvation or physical exercise, you need to switch from anabolism to catabolism. And then to do that, actually you promote a phosphatase. I don't have time to tell you how we discovered the phosphatase that promotes TFEB dephosphorylation. And in this condition, when the nutrient absence, it is known, and this was actually demonstrated by the group of David Sabatini at MIT, that mTOR1 it becomes inactive. And then so in this way, TFEB can go into the nucleus and promote catabolism. So you inhibit biosynthesis pathways, and you promote a catabolic pathway. So this is, in fact, what we define as a lysosome-to-nucleus signaling pathway. And it basically regulates the switch from anabolism to catabolism. So we also later discovered that there is actually a feedback loop between mTOR1 and TFEB, because not only mTOR1 regulates TFEB, but we discovered that TFEB itself regulates mTOR1. This was done by a very talented postdoc in my lab, Chiara Di Malta. So we asked, do TFEB levels affect mTOR activity? And the answer, of course, is yes. So when we overexpress TFEB, in this case in vivo, but also in cell culture, this was done by using viral-mediated gene transfer. The activity of mTOR, which is measured, is increased. Ok. Penso che la batteria, forse. The activity of this kinase is increased. And you can see it better here by immunohistochemistry. This is a substrate of mTOR. When we overexpress TFEB, like in this case, this is the control, you get an increase of the phosphorylation of this substrate. So this is the only genome editing slide that I show in order to demonstrate that the mechanism for this regulation of mTOR by TFEB. We discovered actually that one of the targets of TFEB is a RAG GTPase that is important for the recruitment of mTOR to the lysosomal membrane. And this is a direct target, as we demonstrated by chromatin immunoprecipitation. And so to have a final evidence that this is the one that mediates the effect of TFEB of mTOR, we deleted this particular region, so very few bases, from the promoter of this RAG GTPase. And then when we did that, we lost the effect of TFEB on mTOR activity. Yes. As you can see here. So, so basically, now we know that TFEB regulates the RAG GTPases that regulates mTOR activity. And then mTOR has a negative feedback loop on TFEB because it's a negative regulator of TFEB function. I'm only going to show you now, I'll skip this, but I want to show you the impact. So this mechanism, we discovered that it's important for the adaptation to nutrient availability, and you can imagine that this kind of adaptation is very important. It's been very important during the evolution of the species. So we tend to eat quite regularly, but in the wild, animals don't have the ability to do that. So they have to rapidly switch from situation of fasting and the active physical exercise to situation of feeding. And so while in this situation they have to activate catabolism, in this situation they have to be able to switch rapidly from catabolism to anabolism. And we think that the mechanism that we identified is very important for this switch. And we think that this mechanism is also important for in some disease conditions, and particularly in cancer. So I'll only show very few slides on this, 2 or 3 slides on this. So there are some type of cancers, such as melanoma, renal cell carcinoma, alveolar sarcoma, and also pancreatic cancer, that are highly dependent on the induction of autophagy and of lysosome biogenesis. And in this type of cancers, people have found high levels of expression of these transcription factors, TFEB and other members of the same family. And in fact, some patients with the renal cell carcinoma have chromosomal translocation with very, very high level of TFEB or TFE3, and patients with melanoma, in some cases, have very, very high levels and gene amplification of MITF. And so I'll skip this. So we found that there is upregulation of the RAG GTPases that activate mTOR activity in renal cell carcinoma, pancreatic cancer, and melanoma as an indication that the oncogenic effect of this transcription factor is mediated by the increased mTOR activity, in addition to increased autophagy. So actually there is a contradiction, because situations where autophagy is increased, mTOR activity is low, because you want, in those situations, autophagy is catabolism, mTOR is anabolism. But in cancer cells, in some type of cancers, both processes are activated, because this is kind of a nutrient scavenging effect. They take all they need in order to promote their growth, because they are very actively proliferating. And so, and we found that rescuing of the mTOR1 hyperactivation will limit the hyperproliferation of cancer cells. And so this is actually the final experiment. We did this in collaboration with the group in Milan, the group of Pier Giuseppe Pellicci and Luisa Lanfrancone. So we took melanoma cell lines with very high levels of MITF, which is the equivalent of TFEB in pigmented cells. And we injected in mice, and they induced tumors of this size. But then when we silence the RAG GTPases that is induced by MITF in these cell lines, then you see that the tumor size and number is significantly reduced. And this was published in Science last year. So I hope I convince you that the lysosome is not a simple trash can, but in fact can be viewed also as a control center. So it's not only the last step of a catabolic pathways, but it's also the initial step that controls many others, such as cellular energy metabolism. So the lysosome, in fact, mediates intracellular clearance by autophagy, lysosomal exocytosis, and so degradation and recycling processes. And so this is important to clean the cell. And it's important, for example, in disorders such as neurodegenerative disorders. It's important to promote its activity. But an excessive autophagy and lysosomal function has side effects. In fact, the lysosome also mediates signaling. It's like nutrient sensing, response to starvation, and to physical exercise, and switch between anabolism and catabolism. And if this effect is enhanced, then it promotes also, unfortunately, cancer proliferation and cancer growth, because it promotes the metabolism of cancer cells. So most of the experiments the data that I discussed were produced in a very nice institute, which is not too far from here. It's in Pozzuoli. And this is actually a photo of the institute. And you're all invited to visit TGEM, the Teleton Institute of Genetics and Medicine. And also, so there, of course, in the institute and in my group, there's many talented people, very talented young people. The data that I discussed were produced, the most recent ones, by Chiara Di Malta and also by Gennaro Napolitano. We are very lucky to get the support of the Italian Teleton Foundation, but also from several other important international funding agencies. And I also already mentioned some important external collaborators. And I like to end with this advertisement. There is a meeting which is on a topic that it's quite overlaps quite well with the topic of this meeting. It's the 6th International Conference on Undiagnosed Disease of the Undiagnosed Disease Network. Is basically focused on the genomic sequencing to make diagnosis of patients that have been they've gone around the world to find their diagnosis, and they don't know what they have. And usually by doing genomic sequencing, according to our records, in 50% of them you're able to make a diagnosis of, and sometimes these are new disease entities. And so we actually run the Italian Undiagnosed Disease Project in our institute, which is something that we like very much. Thank you very much.